Good morning, everyone. Welcome to lecture 3 of BMS 300. Can you all hear me at the back? No? Let's go a little higher. That'll be, I think, extra credit question. What is the first question after my class at the start of class? Can you hear me? Alright, so today we'll be talking about, is this better now? Not yet? Not better? Oh. Is it any better now?
Maybe you could just scream.
Good, okay. So, as we proceed in the lecture, if you still can't hear me, don't worry about interrupting the class. Just say, could you speak louder or increase the volume, we'll do something, alright? Because it's no point you sitting there and listening to me if you can't hear what I'm saying. Sounds good? Alright, so today we'll be talking about the lipid bilayer and proteins. But before we do that, I gave you a few polar questions to work on yesterday after class, and so we can practice that now, alright? And so read the question carefully. There may be something different, so just don't blindly select the answer we discussed yesterday, right? So here's the question. You have a beaker with two chambers. On the left side you have 15 millimolar urea on the left, and on the right you have 30 millimolar of NaCl, and both are separated by a membrane which is permeable only to water. Which of the following do you think can occur? Do you think water will not flow? Do you think water will flow from right to left, or from left to right, or do you think it's none of the above? Yes.
Is there, do we get points for this?
No.
Okay, because it was like register with your percentage and get credit.
Oh, no, there's no points for this. It's just for my understanding that you are, for my understanding of your comprehension. How about that?
Okay, just making
Yeah.
sure.
All right, we have 78 responses. Let's see what you said. Most of you said water will not flow. The correct answer is actually water will flow from left to right. The question we discussed in class yesterday is not the same as this question. All I did was I changed urea from one thing to NaCl on the other side. So what do you think the right answer is? From left to right. Okay, so just make sure you read the question carefully. These kinds of questions are great for me because all I do is change one word and I get 3 different questions. So I hope you understand why the water will flow from left to right, because on the left side you have 15 millimolar of urea, and that means there's more water molecules compared to the right side where you have 30 millimolar of sodium chloride, which will dissociate into 30 ions of sodium and 30 ions of chloride, and so it will be 16 millimolar concentration. So the water will flow from left to right. Is that clear, folks? Right. So the next question is, which of the following are responsible for hydrophobicity? Is it A, charged particles, B, nonpolar particles, or B, polar particles, C, nonpolar particles, and D, more than one option is correct. Yes?
Can you turn it up a little?
Turn up a little, sure. Is that better? No?
A little bit.
Is it better now?
Yes.
Okay, is it better now?
Yes.
Alright, okay. So, we got 96 responses. So most of you said nonpolar particles, and that is the correct answer. If particles are charged or polar, they can form hydrogen bonds with water, and so that will lead to hydrophilic interactions, not hydrophobicity. So with that, so C is the right answer. The last question is, atoms in a water molecule are linked by which type of bonds? Is it ionic bonds, covalent bonds, dipole moments, or is it by hydrogen bonds? Oh no, it's working now. We have over 100 responses, and the correct answer is covalent bonds, because the hydrogen and the oxygen atoms, they are sharing electrons, and that is a covalent bond. Okay, so with that, let's get into what I hope to discuss today. Here's an overview of this lecture. So today we'll be talking about phospholipids and the lipid bilayer. So this is a representation of what a phospholipid will look like. There's going to be a hydrophilic head and a hydrophobic tail. So you see that it's an amphipathic molecule. And the way these amphipathic molecules are organized in the cell membrane is that it is found in a lipid bilayer, where you have two layers of these phospholipids. Now the importance of this we will see in a few minutes. And notice that along this phospholipid there are a number of proteins which are punctuating this lipid bilayer. And the question we ask is, where do these proteins come from? What are their functions? And so to discuss that, we'll talk about the structure of different proteins, and we'll talk about primary, secondary, tertiary, and quaternary structures. So just keep in mind that the primary structure of proteins is just amino acids linked one to another. The secondary structure is got a little more complexity to it. It could have alpha helix or beta sheets, and if those secondary structures interact with each other in a single chain of amino acids, that can lead to a tertiary structure. And if you have multiple subunits come together, that's called a quaternary structure. Now keep in mind that the function of a protein is highly dependent on its structure. So we talk about the structure-function relationship in both normal functioning and also pathology as well. So here's the structure of a phospholipid. When you look at it, what you will notice is that it is a modified triglyceride. So yesterday I showed you the triglyceride structure. It has 3 carbon atoms. And notice that each of these carbon atoms will be linked to fatty acid chains. So notice here that this carbon is linked to a fatty acid chain here, and the other one is linked to a fatty acid chain as well. But the third carbon, it's linked to a phosphate group, and that phosphate group could be linked to an R group or a remaining group. That R group is most likely charged, and that's why it is hydrophilic. And these are examples of some common R groups, like choline, serine, phenylamine. I don't need you to memorize this, but just appreciate that the group linked to the phosphate is going to be charged. And so we understand that phospholipids are amphipathic because they have this hydrophobic tail and a hydrophilic head. And let's do a thought experiment. Let's say you have a beaker with water in it, and you have a salt shaker with these phospholipids which can be shaken up. And as you start adding the phospholipids to the water, you'll notice that the hydrophilic heads can interact with the water to form the hydrogen bonds, whereas the hydrophobic tails will be pointing away from the water. But let's say you keep adding more and more of these phospholipids, eventually they'll reach a point where they're going to all band together and form this single layer of sphere known as a micelle. And micelles are something we will encounter when we discuss the digestive system. But the thing about micelles is that they are not very stable. All you need is to have one water molecule penetrate this hydrophobic region, and the entire micelle will fall apart. And so this lipid bilayer is going to be more stable, and it's also known as a liposome. And notice that in this arrangement, the inside of the cell is interacting with the hydrophilic phospholipid heads, and also the outside of the cell it can interact with an aqueous environment. And when you think about how these the lipid bilayer is represented in biology, it's always not represented like a sphere, but it's rather represented as this flat planar lipid bilayer. And the reason for this can be explained by considering the dimensions of the phospholipids and also the molecular dimension of the cell. So if you think about the diameter of the phospholipid head, it's around 1 nanometer, whereas the cell's diameter is around 10 micrometers. Now, what this means for us in terms of this planar lipid bilayer model is that you would need around 30,000 of these phospholipid molecules to surround the entire cell if those phospholipids were laid side by side. And so if you consider that the cell is spherical, and it's like 360 degrees, you take 360 degrees and you divide it by 30,000, you're going to get a degree of curvature of, you want to do the math, 360 by 30,000. You all have your phones on you, don't you? So anyone can try that. 360 by 30,000. 0.012, right? So that is going to be the degree of curvature between the adjacent phospholipids, right? Now, from this perspective, it's going to appear flat. And if you think about the real life example of the surface of the Earth, the circumference of the Earth's surface is close to 25,000 miles. So if you take 360 and divide it by 25,000, you will get a value of 0.014, which is pretty close to this degree of curvature. And if you consider, in order to go a single degree along the Earth's surface, you need to travel close to 70 miles. And I hope that explains why we represent this lipid bilayer as a planar model, because of the cell's size and the size of the individual phospholipid heads. Is that clear? Yes? Okay. So in this lipid mosaic model, notice that there are a number of proteins which are found embedded in this cell membrane, and these are known as transmembrane proteins. And these transmembrane proteins have a number of functions, and I'll mention a few of them now. So the first role is that they're going to play a role in intercellular communication. So if something is happening inside the cell and the cell wants to communicate it to the external environment, it can do that by expressing certain proteins on the cell membrane. An example of this we will consider when we talk about the immune system, when there are these macrophages and they can eat pathogens. They will have small pieces of these pathogens shown on the surface of the cell, so that neighboring cells can know how to appropriately react. And keep in mind that this lipid bilayer is going to be a very strong defense against anything which is hydrophilic from entering inside the cell. So if there are ions, they cannot easily enter the cell because they have to get past this hydrophobic region. And so these transmembrane proteins can serve as ion channels through which sodium ions, chloride ions, potassium ions, all of them can move in and out of the cell. And these channels can play a very important role in maintaining the ion concentrations inside and outside the cell. And we will see the application of this when we talk about the nervous system. If a specific ion concentration is not maintained, it can prevent the neurons from firing, and that can have an impact on the muscles which they activate. So notice that these proteins can also have sugars attached to them, and that's called glycosylation, the attachment of sugars. And we will see how this is done when we talk about the processing which goes on in the endoplasmic reticulum and also in the Golgi apparatus. But for now, just know that this can play a few roles, such as helping with the extracellular matrix attachment. It can also modify the shape of proteins to help with certain ligands to bind to the protein because of these sugars linked to it. And finally, it can also ensure the stability of a given protein. So this can lead to the next question: where do proteins come from? And I will explain that by talking about the central dogma. So according to the central dogma which was proposed by Watson and Crick, notice that the DNA is used to make RNA through a process known as transcription, and the RNA can be used to make proteins through a process known as translation. Keep in mind that this transcription is going to take place inside the nucleus because the DNA cannot leave the nucleus, and so an RNA is generated, and that RNA can be sent out into the cytoplasm through nuclear pores, and that RNA will be used to make proteins in the cytoplasm or in the endoplasmic reticulum, and it can be further processed to perform various functions. So what I'm doing now is I'm going to give you a very high-level overview of how this protein synthesis happens. So it's all going to start with the signal reaching the nucleus, and when that signal reaches the nucleus, the DNA template is used to make an RNA. And what this RNA does is it's got the information specifying which amino acids should be in that specific protein and what order they should be arranged in. And so the various amino acids are arranged in a specific order, and they're all linked to one another, and then that protein can be released for whatever function it has. But consider that you're on a very stringent diet, right, and you don't have a certain type of amino acid in your diet. Do you think your body will make the protein when there's required amino acid missing? Do you think it will do that? Yes? Will it go ahead and make the incomplete protein? Yes or no? Simple one. No. Yeah, the answer is no. Because if it were to make an incomplete protein, that's going to change its structure and the overall function will be affected. So the body would rather just not make that protein if it doesn't have the complement of required amino acids. So this does not mean that for every meal you need to make sure that you have the right complement of amino acids. As long as you have a varied diet once every few days, that should be sufficient. So with that, let's move on to talk about the structure of proteins. So we will be talking about the primary structure of proteins, how they are consisting of just amino acids linked one to another by these bonds known as peptide bonds. It looks like a string of beads. And we'll also see how those amino acids can be either arranged in alpha helical manner or in a beta sheet, depending on the types of bonds which are formed between the various amino acids. And if these alpha helices and beta sheets can interact with each other, they can take on a more complex tertiary structure. And if you have multiple such subunits, so if you have multiple of these amino acid chains coming together, that can form a quaternary structure. And I will give you examples for these. So if you consider the amino acid, right, its general structure is that it has a alpha carbon in the middle, and it also has this amino group and this carboxylic group as well. So every single amino acid will have these three groups. But keep in mind that the main difference between the amino acids is the R group shown here, or the remaining group, and that's what makes one amino acid different from the other. So here are the 20 different R groups which make up the various amino acids. And the good news is I don't want you to memorize these. I don't even require you to know the names or the structures. Those of you who will be taking biochemistry in the upcoming semesters, you will have that pleasure, right? It's not up to me to give to you. But it is kind of like a language, right? You learn the alphabets, helps you understand what it means. But you can classify these R groups into different types. So for example, you have these non-polar R groups, and they are not able to interact with water. You also have these polar R groups which can interact with water. Then you have these aromatic R groups which are made up of these benzene rings. They can't interact with water. And finally, you have charged R groups which can interact with water. So it is enough to say that some R groups are hydrophobic and others are hydrophilic, and that ties into what we were discussing yesterday. there's another side point I want to make, that some of these R groups can have sulfur in them, right? For example, notice that where is it? Cysteine right here has a sulfur, methionine also has a sulfur, and the importance of this I will tell you in an experiment which we are going to discuss on how related to nucleic acids, right? So that's the reason why I'm mentioning sulfur here. So how are these various amino acids linked to other? They're going to be linked by these peptide bonds, and a peptide bond is a covalent bond which is formed as a result of a dehydration reaction. So the OH from this carboxylic group and the H from the amino group, they will be released as water, and as a result you have this peptide bond being formed here, and again this is a covalent bond. So it is through peptide bonds that you have the primary structure of amino acids. So now we'll move on to talk about what causes the secondary structure of these amino acids. So the first one is the alpha helix we'll consider. Notice that the alpha helix is going to look like a very compact corkscrew, right? And so why is it like a compact corkscrew? So let's take a closer look at what is along this this helix. Notice that for a complete turn, it is around 5.4 angstroms, and in that, notice that there are 3.6 amino acids for each turn of the helix. And between each of these amino acids, the distance is 1.5 angstroms. So these are numbers I don't want you to memorize, but I just want you to appreciate that these are the dimensions. Now let's answer the main question: why are these amino acids forming the alpha helix? What is the kinds of bonds which they are forming? So the bond which is formed to form this alpha helix, it's a hydrogen bond, and these hydrogen bonds are formed at a very specific pattern. So this pattern is I+4 hydrogen bonding, where I is referring to a specific amino acid, and it's going to form this bond with another amino acid which is 4 amino acids residues away. So for example, let's say this is amino acid number 1, this is number 2, this is 3, here's 4, and here's 5, right? You saw those 5 numbers up there? We have numbered 5 different amino acids, and because of this pattern, 1 is going to form a hydrogen bond with number 5, because I+4 is 5, 1 + 4 is 5. Now the way this hydrogen bond is formed is that this ketone group C double bond O is going to have a partial negative charge. The oxygen has got a partial negative charge, and so it is going to interact with the hydrogen in this NH group here, and that is how these alpha helical structures are formed. So for example, let's say number 3, residue number 3, which will it form its hydrogen bond with? 7, right? So 3 + 4 is 7, and if it's 2, it's going to form it with number 6. So that's how these hydrogen bonds are formed. Keep in mind that the R groups are not involved in these hydrogen bonds, right? It is only the peptide bonds which are interacting, the atoms of these peptide bonds are interacting with one another to form the hydrogen bonds. And these R groups, they're going to stick outward from the helix. So let's think about the lipid bilayer and transmembrane proteins. Now let's say this alpha helix is embedded in the lipid bilayer, all right? What type of R groups do you think that alpha helix will most likely have? Will it have hydrophobic residues or hydrophilic residues in the R group if it's the middle of a lipid bilayer? Hydrophobic or hydrophilic? Hydrophobic. Why would it be hydrophobic? Because the majority of the lipid bilayer is going to be made of that hydrophobic tails, right? And that's why the alpha helix R groups are going to be hydrophobic, so that they can interact with that majority of the lipid bilayer. Is that clear? Okay, go up higher. Okay. So what's the last thing you heard before my voice fell off? Your radar.
The 4 + 1 = 5.
Okay, all right. Okay, wow. I have to rewind quite a bit back. So I was saying that in order to form the alpha helix, the way the hydrogen bonds are formed is according to this pattern here, I plus 4. So residue number 1 is going to form a hydrogen bond with residue number 5 because it's 1 plus 4 is 5. Are you clear with that? Yes? Okay. And then the next thing I discussed was how is this hydrogen bond formed? It's formed between this negative charge of the oxygen in one of those peptide bonds along with the positive charge of the hydrogen. That's how these hydrogen bonds are formed. Okay, that was the second thing. And thirdly, I was asking what type of R groups do you think will be found in the alpha helix if it was in a lipid bilayer? And the question was, is it hydrophobic or hydrophilic? And the answer was hydrophobic because the R groups are sticking out and they should be able to interact with that hydrophobic region of the lipid bilayer. Is that clear, folks? Yes? Okay. So moving on to beta sheets, we see that the main difference between alpha helix and beta sheet is that the alpha helix is very compact, whereas the beta sheets are more extended. And the reason why they are more extended is that even in beta sheets, you have the hydrogen bonds formed between those peptide bonds. But the main difference is that it's not formed following a very strict pattern like I plus 4. In fact, when two peptides are lying side by side, it's going to form those hydrogen bonds. And that is the reason why these the beta sheets are not having a very compact shape. Another difference between the alpha helix and the beta sheets, which are not listed on the slides, is the positioning of the R groups. In the alpha helix, they're going to point outward. In the beta sheets, they're going to point upward and downward. Is that clear? So the orientation of the R groups will be slightly different between the alpha helix and beta sheets. Is that clear, folks? Yes.
Which way do they face?
So in the beta sheets, they'll be up and down. Yeah. Because it's like a sheet, and so the R groups will be pointing up and down. If there are no further questions, can we move on to the tertiary structure of proteins? Yes. Okay. So the way tertiary structure of yes.
Why did the R groups point upward and downward for the betas?
Because I didn't get into this because the peptides could be in parallel arrangement or anti-parallel arrangements. So it could, if it's going back and forth like this, then it could point upward and downward. Yeah. Good question. So the question is, why does it point up and down and not outward? So when you consider the tertiary structure of proteins, this is going to be mainly influenced by hydrophobic and hydrophilic interactions. For alpha helices and beta sheets, it was hydrogen bonding. For primary structure, it was peptide bonds. For tertiary structure, it is hydrophobic interactions. So consider that this is a peptide chain, and the polar groups are shown here in blue, whereas the green groups are denoting non-polar R groups. So non-polar R groups, they don't want to interact with water, and so they are going to form the core, whereas the hydrophilic groups, they're going to form the exterior of the protein. So that's how the tertiary structure can be brought about. Now, the quaternary structure is where you have multiple subunits coming together, and we'll see this example in hemoglobin, and that's what you call as a quaternary structure. So you just don't have one single long chain, but you have multiple chains coming together to form a complex structure. So now let's look at some of the examples of protein functions. So we see that proteins can play a role as receptors, as hormones, which bind to those receptors. They can also be enzymes which catalyze reactions and make them go faster. And finally, they can also be transport proteins. And all of these functions I can highlight by using this example of how insulin can permit glucose to enter into the cell. So if you notice this cartoon here, insulin is shown here as a purple circle. It is a hormone which is made up of amino acids and so it's an example of a protein. And the receptor to which it binds, notice that it has multiple subunits. It's got 2 alpha subunits, 2 beta subunits. So it is a protein which has a quaternary structure, and this is the insulin receptor. And notice that in this beta subunits, there are these P groups attached, and the P is referring to phosphate groups. In order to add a phosphate group, you need an enzyme known as kinase to do that, and so the kinase is also a type of protein. And finally, this is going to trigger this P13K signaling pathway, which is beyond the scope of our discussion. But the outcome of that signaling is that this vesicle, which has this glucose transporter, is going to fuse into this lipid bilayer, and glucose can bind to that transport protein and then be moved inside the cell. So I've shown you how proteins can do all of these functions by using this example. Proteins also are used to make antibodies, and antibodies are what are made by our immune cells to target specific antigens or pathogens. And these antibodies have a very specific antigen binding site, and so it is fine-tuned to bind to specific antigens, and we will see how this is brought about when we discuss the immune system. So now let's talk about what is going to determine the shape of a protein. So we know that the types of amino acids which are in a given protein chain will play a role, and also the order in which these amino acids are being linked to one another will also play an important role, because depending on the order of these amino acids, those amino acids can interact with one another. So the third thing is the bonds which are formed between these amino acids. Now let's look at a few examples of these bonds. First one is an example of a disulfide linkage. I showed you that there was an amino acids which have sulfurous in them, so they can form this disulfide linkage. Another example is the formation of ionic bonds, and we know that some amino acids are positively charged, others are negatively charged, so they can have an electrostatic attraction between them. And then you have these hydrogen bonds formed as shown in the case of both alpha helices and beta sheets. And there could also be hydrophobic interactions because there are a number of non-polar amino acids. Final property which can affect the protein structure is also the environment. So proteins can be extremely fragile, and so if you were to change the environment by either increasing the temperature or the pH, that can have an impact on the structure of the protein and eventually its function as well. So to highlight the importance of the environment and its influence on protein structure, I want to show you the example of a raw fertilized egg. Right? If you break open a fertilized egg, it's got this egg white which has mainly albumin, and it also is the yellow part, the egg yolk, which is got a number of proteins. And all of these proteins, their goal is to help this fertilized egg eventually become a chicken. Right? So all these proteins are inside the egg. Now my question is, what do you think would happen if we got rid of the shell? Right? Do you think the chicken can still be formed without the shell? Right? So just take a minute to answer this whole everywhere question. Yes or no? Do you think removing the shell can Ooh, we got 100 responses. Oh man, what is this? 58 said yes, so the majority seemed very optimistic, and some said no. Alright, okay, let's see what a group of Japanese high school students were able to do as a project. Oops. It's in Japanese, so the subtitles are there. So they stick it in an incubator. Alright, so I guess the 58 of you were right. So, but the thing is, coming back to our point of maintaining an appropriate environment, in this experiment they had incubator. Right? An incubator is to always have to maintaining the temperature and the humidity. And I took a look at this journal article they published, and the temperature at which the incubator was set was around 99 degrees Fahrenheit to 102 degrees Fahrenheit. So it was very tightly regulated. The temperature was tightly regulated. Now let's assume that there was a power outage, and something goes wrong, and the power comes back on, and instead of the temperature being maintained within that narrow window, it jumps up to 130 degrees Fahrenheit. Not too much, just 130 degrees Fahrenheit. They would not have chicken in the lab. They would end up making breakfast, right? So all of you agree with me that this can turn into a live chick. So, and this fragility of proteins is something which is used by chefs to make some pretty tasty dishes like meringue and so on, right? So the shifting gears to talk about some pathology examples, can there be harmful proteins? And yes, we will consider prions. So prions can lead to a condition in sheep called scrapie, and this is a neurological problem. It's going to affect muscular function, and so you notice that there will be a itching sensation, and so the sheep will try to scratch themselves against the fence, and that can lead to loss of wool. And because the nerve function is affected, you also affect the function of the muscles, and notice that there is an unsteady gait as well. And even in other animals, it can lead to mad cow disease, also known as bovine spongiform encephalopathy. Now the term encephalopathy is a disease of the brain, and it's called spongiform because you notice these vacuoles which form a very spongy appearance in the cortex of the brain. In humans, there's a condition known as Creutzfeldt-Jakob disease, and also there's Kuru, which was prevalent among the people of Papua New Guinea, especially the Foray tribe. This was around 50 or 60 years ago. So this was a disease known as the laughing death because the patients would often have pathological outbursts of laughter. And just like in scrapie, they would have muscle weakness. And notice that these 3 children, they have, or 4 children, they have difficulty even standing up straight. And you can notice that they have this smile on their faces. There's these pathological outbursts of laughter. Now, this was actually linked to their cannibalistic ritual. So whenever a member of the tribe died, the tribe would eat that member's corpse because they wanted that member to always be a part of them. Now the problem with this type of ritual is that if someone died of of Kuru, they will be ingesting those prion proteins and it will keep propagating. And it was interesting to see that this was prevalent mainly among women and children because in a patriarchal society, it's easy to imagine how the men would get the muscle tissue and the women and children would be left with the brain and the spinal cord. So this was this disease eventually got eradicated once the cannibalistic ritual was abolished. So what are these prions, right? So prions, they are proteins which are found in the nervous system, and we are not really sure what their function is. There is some indication that the prions shown here by the C subscript referring to the normal prions, they may play a role in cellular signaling, but we're not sure about their function. But let's say from the environment, you just have this abnormally shaped scrapie prion entering the body. This scrapie prion is denoted by a subscript SC to denote it's abnormal. It's got a slightly different shape compared to the normal prions, and because of this different shape, there are no enzymes in our body which can digest those scrapie prions, and it is very resistant to digestion. And when these scrapie prions interact with the normal prions, it will cause them to change their configuration, and this just keeps spreading. So even the normal prions become scrapie prions, and they will start aggregating until they form these structures called amyloids. Right? And amyloids are found in a number of neurodegenerative diseases. This could be the case with spongiform encephalopathies, Alzheimer's disease, Parkinson's disease, and even Huntington's disease. And this can eventually lead to a condition known as amyloidosis. And I have this video from the Mayo Clinic. I'm not sure that if you could hear the audio if I play it for you. Let's just see.
Briefly, amyloidosis is the
Can you hear it?
depositing of abnormal proteins in your body. All the proteins in your body are biodegradable and recyclable, but amyloid proteins cannot be broken down by your body, and as a consequence,
Can you hear it, folks? Not really. Okay. I'll let you watch the video on your own time, but just know that there are going to be these insoluble proteins which are going to aggregate in different parts of your body, and as a result, that can cause those various organs to function poorly. And so the Mayo Clinic is trying to come up with methods to prevent those insoluble proteins from forming or to cause them to be soluble. Right? So that's what that video was about. But in summary, let me just say what we discussed, and as we did yesterday, we will go through the key questions after we're done with the summary. So we talked about the lipid bilayer, which is the way in which the cell membrane is organized, and that is going to highlight the importance of amphipathic interactions. We talked about amphipathic interactions yesterday in terms of soap and how it can be a degreaser, but today we're seeing how the amphipathic interactions play a role in the cell. And we noticed that there are a number of proteins which are embedded in this lipid bilayer, and that led us to our discussion of the structure of proteins. And we see that protein structure can be determined not only by the amino acids, but their order, the types of bonds between those amino acids, and also by the external environment. And I gave you that, and I used the explanation of the fertilized chicken egg, and we see that even in terms of pathology, the structure can affect the function of the protein. And we talked about Kuru and other neurodegenerative diseases. So that let's spend a few minutes talking about the key concept questions. So this is a question from lecture 1, right? What are the major organ systems and what are their components and major functions? So what is the first system I told you about? I'm pointing to it. Integumentary system. It's made up of the skin, hair, nails. Main function? Protection. All right. And what about the function? What's the skeletal system? Main function? Yeah, support and also bone cell formation. And it's made up of skeleton, the bones, cartilage, tendons. What's the third one? Endocrine. Endocrine. Its function is hormonal signaling. The fourth was inside nervous system, made up of the brain, spinal cord, and the function is to neural signals, detect changes in the environment and respond to that. And what was the next? Muscular system. We talked about 3 kinds of muscles: smooth, skeletal, and cardiac. And the function of skeletal muscle is to ensure movement, and cardiac muscle is to pump blood, and the smooth muscles help to carry the blood to various parts of the body. And we talked about the components of blood. What is related to the defense of the body? White blood cells, and that's made in the lymphatic system or the immune system. And we also talked about gas exchange happening in the respiratory system. And what does the digestive system do? absorb nutrients for the blood to carry to the various parts of the body, and the blood is purified in the renal system or the urinary system, and finally we discuss the reproductive system. So this is just a high level understanding I hope you have for exam 1, right? So what are the structural classification of proteins and how do they differ in complexity from one another? So what are the 4 classifications in terms of structure? Primary, secondary, tertiary, and quaternary. Right? And the thing is, what are the bonds which hold primary amino acids in the primary structure? Peptide bonds in the secondary structure, hydrogen bonds, tertiary structure, hydrophobic interactions, and in the quaternary structure it could be, we never got into it, but it could be hydrophobic interactions as well. And what are the determinants of protein structure? Amino acids, their order, the sequence of amino acids, and the bonds between them, and finally
Types.
the external environment as well that can influence it. And then what are 2 examples of diseases of abnormally shaped proteins? We talked about Kuru, yes? Creutzfeldt-Jakob disease, Huntington's, and what else? CJD, Creutzfeldt-Jakob disease, yeah. Yeah, that's great, folks. So we still have 3 minutes, and I'll end class early, but please go through these questions, and when you come back tomorrow, we'll try answering them in class, right? Have a great rest of your day.